All right, all right, all right. Time to get started. We have a molecule of mRNA that we were able to produce through transcription of DNA. Now, what do we do with it? Well, the messenger RNA molecule is going to make its way outside of the nucleus and eventually find its way to the ribosome. Now, believe it or not, ribosomes are actually made mostly of something known as ribosomal rRNA, right? So the R stands for ribosomal. So here we have our rRNA. And so the rRNA is what makes up a majority of the ribosome. So technically speaking, the mRNA leaves the nucleus on its way to find its little buddy, rRNA. Now, eventually, the message will be read by the ribosome. And the ribosome will then need the building materials or the raw materials needed to synthesize the protein coded for an mRNA. And those raw materials, the amino acids, are actually transferred to the ribosome through or on a molecule of tRNA. Okay, so we've got messenger RNA, right, which typically in my drawings is just kind of a long single strand. And again, it would have that transcribed code from the gene and DNA for whatever protein we're interested in. And then in my diagrams, let's use blue to represent our ribosomes. And you remember in our little diagrams, they were typically just like little dots. So pretend like we're zooming in, okay? And I'm just going to make it a big circle for our RNA. This is our big old ribosome, the site of protein synthesis. So we have our instructions, our message. What protein do we make and how do we make it? We've got our little protein making factory, the ribosome, and now we just need the raw materials. And in my little cartoon drawings, I'm just going to make them little triangles, right? And these little triangles are carrying some really, really important cargo. And let's make them purple. And these would be the amino acids. So the messenger is exactly that. Contains the message, which protein to make and how to make it. The ribosome is like the factory, the protein synthesis factory. It's going to read the message, right, and start to construct the protein. And the tRNA, the transfer trucks, are going to transfer the appropriate amino acid to the ribosome. So you could even kind of think about it as an analogy, right? So here is our car factory. And obviously, I didn't do so well in art. Okay, so here is, I'm using the same colors, right? So here is our ribosome, our little factory, okay? And the factory needs blueprints. The factory needs to know what to make, and it needs the messenger RNA. Now think about it. I'm sure at the Honda headquarters, they've got the original or the master copy of the blueprints for a Honda Accord, and they're not going to send that exact piece of paper or that exact document to all the factories around the world, right? They're going to send a copy of it. They're going to send a message. So the messenger RNA would be that little blueprint copy. Okay, and so it was transcribed from DNA into RNA, right? And so that is going to enter the ribosome. And the factory, right, is going to use the blueprint to build cars or to build something. And where are they going to get the raw materials to do this? Well, obviously, they're going to radio out to their supplier, and the supplier is going to deliver the raw materials. You think I was born yesterday. I'm like, how do I draw a truck? Is going to be filled with the raw materials, right? And obviously, if we're building cars, it's going to be steel, rubbers, plastic, carbon fiber, and all that jazz. But for us, in this case, our little transfer truck is carrying amino acids, and it's going to deliver those to the factory. And then at the end of the day, the factory uses the blueprints and the raw materials to make a protein. And let's use yellow, kind of climactic, right? And then we manufacture our little sports car. Duh. I don't know who's going to pay for that. All right, so there's our little sports car, right? And that was coded for the messenger RNA. The ribosomal RNA, did the hard labor, the hard work, and it used the raw materials supplied by the transfer truck. I don't know. Maybe that analogy will work. Maybe it won't. But we at least tried. All right, let's go on to the next slide. Now, when you see a blank slide, oh man, Mr. Collins is about to start drawing. All right, so here we have our big cell, and inside of the cell we have the nucleus, right? And inside of the nucleus we have our molecule of DNA, right? Which would be the master copy containing the genes for all the proteins this cell needs. Now, when the time comes and the cell needs that protein, the DNA will be transcribed into a molecule of messenger RNA. The messenger RNA can then leave the nucleus on its way to the ribosome. Now, eventually, our mRNA molecule will find its way to the ribosome, and it will enter the ribosome, and the ribosome is made of rRNA. The R stands for ribosomal. And the ribosome, or the rRNA, I like the R here, will read the message. And as the ribosome reads the mRNA, it's figuring out which raw materials it needs and in what order it's going to be put together. And so the ribosome works together with molecules of tRNA, our transfer trucks. The transfer RNA is going to deliver the appropriate amino acid, right? And I'm just going to abbreviate amino acid as AA, right? The tRNA molecule will bring the appropriate amino acid to the ribosome. And if it's the correct amino acid, if it matches, the tRNA matches the mRNA, then the rRNA will put those amino acids together. And eventually what we start to produce is a really long chain of amino acids known as a polypeptide chain. And we all know that polypeptide chains will eventually become proteins. And so this long polypeptide chain will eventually be released and it'll fold over on itself. And, proteins have all those crazy shapes, and then we'll have ourselves a complete protein. And remember, some proteins are actually protein complexes, and they're made of a bunch of proteins that are kind of like stuck together. So we had transcription, got us started up here and made our mRNA molecule, and then mRNA and rRNA and tRNA work together to build a chain of amino acids, which eventually will become a mature protein molecule. And so you can kind of see this in this diagram right here. Let's pretend like this is the starting point. The long, thin blue is the mRNA. We've got the little red bubbles are ribosome. The little yellow elf hats are tRNA molecules, and they're carrying little amino acids. And so maybe this ribosome is attached here, right? This is like time 0, time 1, so it's like as time goes on. You can see the little ribosome slides along the mRNA molecule, slowly reading the code and calling out to the tRNA for the appropriate amino acid. And you can see the amino acid chains getting longer and longer and longer. And eventually the ribosome reaches the end of the mRNA molecule. It releases the mRNA molecule. It releases this long chain of amino acids, and we have a protein. Okay. Now you might see, what's this down here? Codon and anticodon. What's all this mess about? Well, how does the tRNA know if it is carrying the right amino acid? How does the ribosome know what amino acids are needed and in what order to put them? And that's where the codons and anticodons come into play. mRNA is translated by the ribosome using something known as codons. Codons are a sequence of 3 nucleotides. So the ribosome reads the messenger RNA 3 letters at a time. And each codon, or set of 3, codes for an amino acid. So we can see here's our mRNA molecule. And then here would be the code, right? The actual nucleotides. And if I were the ribosome, I would read them 3 letters at a time. So here's our first codon, AUG. And that would code for methionine. This is an amino acid. And a lot of times, and what we'll do in my class is we'll just simply abbreviate. You just write the first 3 letters, right? So that would be met, and then threonine or threonine. You would simply just do THR and so on and so forth, right? And then arginine, just ARG. And then for stop, what do you think stop is? It's just the end of the instructions, right? And so this would be the end of the line, and that would say, okay, we're done reading this mRNA molecule, and our protein is complete, right? And obviously, all these amino acids would be linked together by peptide bonds in the ribosome. Now, again, I've explained the codons. What in the world are anticodons? Well, the anticodons are found on the tRNA molecule, and they ensure that the tRNA molecule is in fact delivering the correct amino acid. So the codon is read by the ribosome, and to double check or to make sure that it is the correct amino acid being delivered, the tRNA molecule has to match the codon. And anti in science just means the opposite. So the anticodon will be the opposite of the codon. And I want you to take a moment and try to guess. What do you think the anticodon is for AUG? UAC. Now let's take a look. So here we have the nucleus. Inside is the DNA. It's been transcribed into mRNA. The mRNA molecule found its way to the ribosome, this big blue blob, and the ribosome is reading the mRNA, and we can see that it's reading it 3 letters at a time, right? And so when it read this first codon, AUG, it needs to make sure that the correct tRNA molecule, this crazy looking thing, shows up and attaches. And it does this with the anticodon. So this is the anticodon right here for AUG. That's the codon. This is the anticodon. And the anticodon is found on the tRNA molecule. And since those match, this tRNA molecule would drop off this amino acid and the ribosome would slide over. And then it would read UUC, that's the codon. Okay, we've got the right anticodon. The tRNA molecule could dump off that amino acid and then head on to go find another one, and the ribosome would slide over to the next one, AAA, and, well, that looks like a good match to me. That tRNA molecule is probably going to attach. It's probably going to drop off lysine, and the ribosome will continue to slide over until it gets to a stop codon. Now, as molecular biologists and biochemists and geneticists, when we're studying the code of DNA, right, it's pretty easy to first transcribe it, but translating it can be a little more difficult. But scientists have gone and made this beautiful chart. This is called a mRNA codon chart. So whenever you use this chart, make sure that you're using the codon on mRNA and don't use the anticodon. So we always use the mRNA codon with this chart. Now let's practice with this, right? So I'm just going to give you one little example. So here's our molecule of mRNA. Now, you did notice in the last couple of examples, we had a start codon. Now, when you work out examples, they don't have to start with that. We just typically see that in nature. So I'm going to throw you a curveball. Let's say that our first codon on our mRNA molecule is CCC. And then let's do U, C. And then let's do G, C, C. How in the world am I going to read this? Well, I'll show you. We can kind of think about it as a 3-letter codon, and we can think that's the first base, that's the second base, and that's the third base. So to use this chart, you can see they're labeled first base, second base, and third base. All right, so our first base is C. So I look here. Okay, here it is, C. It's going to be somewhere in here. Second base is also C, so I look up here. I said, okay, C. Now where do they overlap? So it's going to be one of these 4. And I know it's pretty easy. I can see it right there, but just entertain me. So we look over here. It's going to be one of these third. Oh, we can go over here and say, okay, there it is. CCC codes for proline. And so I'm going to write PRO for proline. And I like to draw a little circle around it because it reminds me, oh yeah, we're talking about amino acids, right? And so I'm going to make a little peptide bond to get ready for the next amino acid. So let's look. UUC. So I got UC, phenylalanine. And so let's do PHE. And let's make it an amino acid. And let's make our next peptide bond. And let's read our last codon together. GCC. And go down here. GCC. And that's alanine. ALA. And I'll draw a circle. And for our example, for our practice, we're done. Okay? And so it's that simple. Now, it does take some practice. And there's a reason I had you do the first and second and third base, because sometimes these diagrams won't have the 3-letter codons up here. They'll just have the amino acids, so you have to be able to read them to be able to figure it out. And you can also see that there are stop codons, right? Now, I will tell you, anytime you're practicing, again, don't fall victim. Don't assume that it's always going to start with methionine and it's always going to stop with stop. For practice problems, right, it can be anything. We're testing your ability to take this codon, the 3 letters, and to translate it into the appropriate amino acid. And I will make a video with some practice problems and I'll work them out for you. And so you can check that out, and we'll go over DNA replication practice, transcription practice, as well as translation practice.